Good morning, students. So, last class about the pigments we have discussed. So, in the plant pigments, chlorophyll A, chlorophyll B, carotenes, xanthophylls are grouped under different classes we have discussed. So which type of the light does the plant absorb for photosynthesis? So that is nature of light. So keep the heading nature of light. So what are the different wavelengths? So generally the electromagnetic wavelengths of the light, radiations, or electromagnetic radiation, that is visible rays, visible light, cosmic, all these are electromagnetic radiations. Visible light, cosmic rays, gamma rays, UV, visible light, cosmic, alpha, gamma, UV, IR also, IR. So all these are electromagnetic radiations. So in this, the visible light is absorbed for photosynthesis. Visible light is absorbed for photosynthesis. So in that, if you take the range of wavelength of the light, okay, radio waves also we can include radio waves. So in the visible light, the range is 390 nanometers to 763 nanometers. 390 to 763 nanometers is the range of the visible light. So in this, if you take below 390, it is called as UV rays. So below, what is below 760? All these, the UV rays will be below 390. IR rays will be above 763. IR rays will be above 763. So therefore, less than this, less than 390 UV, more than 763 IR. So, but this is visible. But this range is visible light. Okay? So, if you convert it into angstroms, 3900 angstroms, or you can call it as 3900 angstroms, or 7630 angstroms. 7630 angstroms. So, what is visible light? With GR, it includes with GR. Visible light includes violet, indigo, blue, green, yellow, orange, red. So they have different wavelengths, different ranges. So if you take violet, so this is from 390 to 430 nanometers. The range is 390 to 430. Next, indigo, 430 to 470 nanometers. 430 to 470 nanometers. Next, blue 470 to 500 nanometers. 470 to 500 nanometers. Next, green 500 to 560 nanometers. Green is 500 to 560. Yellow, that is from 560 to 600. 560 to 600 nanometers. Next, 600 to 650 is orange. 600 to 650 is orange, whereas red is 650 to 760. Red is 650 to 760 is red. So all these are the different wavelengths of the ranges of the light. So 650 to 760 is the red. So therefore, this wavelength out of which the blue light and red light is used for photosynthesis. So which light is absorbed, which wavelength of the light is absorbed for photosynthesis means it is 470 to 500, 650 to 760 is the range of wavelength for photosynthesis. Okay. Greater than 700, if you see, or 760 or greater than 700, strictly there is a sharp decrease in the rate of photosynthesis. So such kind of energy is insufficient for the photosynthesis. Greater than 700, that is called red drop. So what is red drop? Sharp decrease. Red drop, it's nothing but sharp decrease in quantum yield in the quantum yield at wavelength greater than 760 nanometers. At wavelength greater than 760 nanometers, or 700, you can write it as 700, greater than 700 nanometers, and insufficient energy to drive any part of photosynthesis. Is an insufficient energy to drive any part of photosynthesis. So this is called as a red drop. Okay, right. Next. So we have discussed about the wavelengths of the light. So next, absorption and action spectrum. What is absorption spectrum and what is action spectrum? Let us see. Absorption and action spectrum. Absorption and action spectrum. So what is absorption spectrum? Let us see first. Absorption spectrum means, the name itself says absorption, which light it is absorbed. That is nothing but wavelength of the light. So absorption, it gives information. Absorption spectrum gives information about absorption of light. That is wavelength by pigments. Absorption of the light by pigments. So it is measured by, the absorption can be measured by spectrophotometer. So which way pigments absorb which wavelength of the light, till now what we discussed, that is can be determined by spectrophotometer. Okay, right. So here in spectrophotometer you can easily detect the wavelength which is absorbed. So they found out that the lesser wave transmittant light, that means here if you see, the light is absorbed the light is passed, and here you are keeping the pigments. One pigment you will keep, and you check out the light which is passed. So here some of the light is absorbed, and once it is comes out, it is detected by detector called galvanometer. So galvanometer detects the light. What is the amount of the light which is absorbed by the pigments, and what is the amount of the light which comes out. So then you can know clearly how much amount, what is initial amount, initial light. Initial light, this is what final light. So initial minus final, if you see, we can clearly understand how much amount of the light which is absorbed. So the decrease here, when they observed less light which is transmitted in blue and red light, that means it indicates there it is more absorption. If an absorption is more, less amount of the light it comes out. So that means during the blue and red light, the absorption range will be more. Blue and red wavelength of the light. When you pass blue and red wavelength of the light, so when blue and red wavelength of light is passed in spectrophotometer, then the less transmittance, more absorption of light, and lesser transmission occurred. Okay, right. blue and red wavelength of the light is passed in spectrophotometer, what happens? Because this is the light which is for photosynthesis, therefore only it absorbs more. Pigments absorb more light of blue and red. So therefore that is for photosynthesis. Green light. So vice versa with green light. So when green light is passed, then it is vice versa. Vice versa means there is less absorption of the light and more transmittance. Less absorption of the light and more transmittance. That is what we see in the green light. So green light is completely absent for photosynthesis. It is completely zero. So in green light, the photosynthesis rate is completely zero. In other range of the light, if you can see some photosynthesis, but green light is completely photosynthesis absent. Okay, right. This is about the absorption spectrum. So the name itself says absorption, light is absorbed. Okay, absorption of the light. Next, action spectrum. Action spectrum. Action spectrum. Here it shows action spectrum is nothing but the rate of photosynthesis. It shows rate of photosynthesis at different wavelengths of the light. Action spectrum shows rate of photosynthesis at different wavelengths of the light. Rate of photosynthesis at different wavelengths of the light. So here the light which is absorbed by the pigments in the chloroplast and release oxygen. So continued oxygen bubbles, the release of oxygen bubbles continuously. The release of oxygen bubbles continuously or evolve oxygen bubbles continuously, which is measured by Ordus apparatus. Or by RQ, or can be measured by RQ. RQ means respiratory quotient. Okay. Respiratory quotient. So this can be measured by Ordus apparatus or RQ. Then you know how the rate of photosynthesis is taking place by the evolution of the oxygen which is evolved. So here you get a peaks. So based on the range of peaks, that is when you take the x-axis and y-axis. Here light absorption, here photosynthesis rate. Photosynthesis rate, and here it is wavelength. So when you take Vibgya colors, at a range of blue it is more, completely zero, more, red it is more. That means at blue and red, that means the wavelengths of blue and red, the rate of photosynthesis is more. So this is previously in absorption spectrum wavelength. If you take you get in absorption spectrum same thing, but there you see chlorophyll A and chlorophyll B, chlorophyll A and chlorophyll B. There also at that particular wavelength you see there is absorption, chlorophyll A and chlorophyll B. Here in the x-axis, y-axis, x-axis and y-axis, if you take light absorption and wavelength on the x-axis. Okay, right. So when you combine both the spectrums of absorption spectrum and action spectrum, when you combine both the absorption spectrum and action spectrum, then the graphs also, if you coincide, it exactly matches. At that particular wavelength of blue and red, pigment absorption of chlorophyll A and chlorophyll B will be more. So both spectrums are compared, continue. When both the spectrums are compared, and notice that wavelength of light, that wavelength of light mainly absorbed by chlorophyll pigments, mainly absorbed by chlorophyll pigments, and noticed or also stimulates, you can write, also stimulate the highest or maximum rate of photosynthesis, maximum rate of photosynthesis in efficient manner. Maximum rate of photosynthesis in efficient manner. So maximum rate of photosynthesis is blue and red regions are absorbed by. So finally the maximum rate of photosynthesis is at blue and red regions, and absorbed by chlorophyll A and chlorophyll B, absorbed by chlorophyll A and chlorophyll B in maximum amounts. In maximum amounts. Okay, right. So this is about the action spectrum and absorption spectrum. Action spectrum and absorption spectrum. So what we studied in action absorption spectrum? Action spectrum, the rate of photosynthesis. Absorption spectrum, which pigments which are is absorbed at what wavelength of the light. Okay, right. Next, light harvesting complex. Keep the heading light harvesting complex. LHC, light harvesting complex. LHC. What is this light harvesting complex? This complex contains the photosystems, because these are generally where does pigments are located? In the thylakoid membrane. So pigments are located in the thylakoid membrane. Pigments are located in the thylakoid membrane. So here if you see this light harvesting complex contains about the light harvesting complex LHC, or we also call it as photosystem. It is also called photosystem, PS, photosystem. The light harvesting complex or photosystem consists of it has LH, light harvesting molecule antenna. Light harvesting molecule antenna and reaction center or photocenters. Reaction center or photocenters. Reaction center or photocenter. That is different in different. This reaction center varies in different photosystems. So second part of the light harvesting complex is reaction center or photocenter, which consists of chlorophyll A. Chlorophyll A. So here the the reaction center varies, that is different in different photosystems. Varies in different photosystems. That means the reaction center is at the reaction center is replaced. Suppose the light harvesting center, if you take the light harvesting center in the light harvesting complex or photosystem, here the RC will be, reaction center will be at the center. That means chlorophyll A, which indicates it is chlorophyll A, surrounded by light harvesting molecules. Light harvesting molecules antenna. So antenna is nothing but, what is this antenna? This is the other pigments of chlorophyll B, chlorophyll B followed by the carotene and xanthophyll. All these are antenna. These are antenna. Hundreds of axillary pigments. These are hundreds of axillary pigments. Chlorophyll B, carotenes and xanthophyll are the axillary pigments which are called antenna. These are absorbed. First the light is absorbed by these antenna. And reaction center is nothing but it consists of chlorophyll A at the center. It consists of chlorophyll A at the center. So the light harvesting complex, first the antenna absorbs light and passes to reaction center. You can note down. First the light initially the light is absorbed by the antenna and transfer to the and pass to reaction center. So chlorophyll A in photosystem 1 is structurally different from chlorophyll A. Here if you see, this LHC is of two photosystems. Broadly, see these are inside they are present, two types. The LHCs are of two types. The LHC is of two types. That is LHC 1 for photosystem 1. We call it as photosystem 1, that is at wavelength 700, and photosystem 2. And it has photosystem 2, that is P680 wavelength of the light. So two types, photosystem 1 and photosystem 2. So in both the cases, same reaction center, chlorophyll A, everything is present. And what the difference? Why the wavelength is different means the chlorophyll A in photosystem 1 is different. Chlorophyll A in photosystem 1 is structurally different from, you can write structurally, is structurally different from chlorophyll A of photosystem 2. So photosystem 2 chlorophyll A is different from photosystem 1 chlorophyll A. That means here I told you chlorophyll A is present. The chlorophyll A of photosystem 1 can absorb light of 700 wavelength, whereas the chlorophyll A of photosystem 2 can absorb of 680. That is why two types it is, that is photosystem 1 and photosystem 2. So chlorophyll, therefore chlorophyll is the chlorophyll A is the chief pigment. Chlorophyll A is the chief pigment or primary pigment in photosynthesis. So it is a chief pigment or primary pigment for photosynthesis. Okay, right. Next, The next is, so this is about the LHC light harvesting complex. So next we have to discuss about the photophosphorylations, that is light reaction. So we are left with light reaction, photophosphorylation is of light reaction, and in light reaction cyclic and non-cyclic. So what is cyclic photophosphorylation in light reaction, and what is a non-cyclic photophosphorylation? So that we have to discuss. Okay, so keep the heading light reaction. Light reaction. So in light reaction, two types, cyclic and non-cyclic. Cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic photophosphorylation and non-cyclic photophosphorylation. So what is this cyclic? The light reaction means, we already know, we studied what is light reaction. Light reaction means the amount of the light which is absorbed, that is it converts the light energy to chemical energy. So in light reaction, the light energy is converted to chemical energy. So that is ATP, in the form of ATP and NADPH, assimilatory power. So how does it converts? Is by cyclic and non-cyclic. Cyclic and non-cyclic. So formation of energy. What is photophosphorylation meaning? Photophosphorylation means formation of ATP phosphorylation, formation of ATP and NADPH, which is nothing but the assimilatory power. Which is the assimilatory power. So formation of ATP and NADP, the assimilatory power during, by using light. By using light in photosynthesis. That is called as a photo. Photo means light. Phosphorylation means synthesis of ATP or energy. So light, in presence of light, energy is produced. We call it as photophosphorylation. Okay. So such kind of phenomena we see. So this, so we have to discuss about the cyclic photophosphorylation first, and then non-cyclic. Okay, right. So in the next class we will see what is cyclic, very important aspect, and what is a non-cyclic. Okay, then here as the pathway occurs in cyclic, so wherever the electrons are moving, again they come back to the same reaction center, we call it as cyclic. If they don't come to the same reaction center, we call it as non-cyclic. So how does that movement takes place? That we will discuss in the next class. Okay, right. So thank you.